Hello, my name's Dr. Harvey Woodworth. I'm a scientific advisor to MEBO research. I would like to just today share with you a presentation on dimethylsulfide. This presentation is complementary to a scientific paper I wrote in April, and this paper constituted a literature review of dimethylsulfide, looking at both its behavior in physiological systems and also those pathoses where blood-borne presence of dimethylsulfide is a feature. The reasoning behind approaching this topic was following the work of a European group of researchers, beginning with a paper they published in 2007. This group identified dimethylsulfide as the main volatile implicated in the vast majority of cases of extraoral blood-borne halitosis. To discuss these concepts, we are going to have to do a little bit of background, cover some definitions, and go through some very basic physiology. Halitosis could most simply be defined as any perceptible malodor on the breath. The study of this area of medicine is known as halitology. As we all see, this subject spans a large number of conditions. This is a schematic of the classification system that has become accepted. And the main point here is that, as with so many other things, halitosis is a symptom and not a diagnosis. The primary subdivision is between genuine halitosis, where there is an actual malodor detectable on the breath by whatever clinical examination or investigation is employed, and with so-called pseudohalitosis, where there is no genuine malodor for whatever reason the patient is experiencing it. Pseudohalitosis is a complex area. The main specialities involved are neurology and psychiatry. Essentially, we're looking at two types of disorder. The first, where there's been some neurosensory disruptions, the olfactory apparatus. An extreme example of this would be a central space-occupying lesion, compressing the olfactory bulb or tract, and distorting the perception of odour. This is an example of a symptom known as cocosmia, sometimes seen with brain tumours. The patient experiences an apparent foul odour continuously, and perhaps this feature can be used to differentiate between cases of genuine halitosis and those of pseudohalitosis. Where the malodor is genuine, the patient's olfactory system is working as intended. There's no defect with it. And a trait of how the olfactory sense operates is that it quickly becomes fatigued or exhausted. A continuous stimuli will lead to the brain disregarding it after mere minutes. And this leaves genuine halitosis patients unable to self-detect their own symptom. The other types of conditions that can cause pseudohalitosis are somatoform and psychogenic disorders, where the symptoms are created within the patient's mind, or the patient is exhibiting an oversensitivity to what is normal, to what is physiological. Olfactory hallucinations are also known to occur in certain conditions. For example, schizophrenia. Where the halitosis symptom is genuine, this can be thought of as either physiological or pathological. It could be considered normal to an extent to have a perceptible odour on the breath, and this is especially true upon waking, largely due to decreased salivary flow during sleep and consequent oral stagnation and increase in oral microbial activity. Colloquially, this is known as morning breath. Another example of physiological halitosis would be consumption of volatile foodstuffs, which leave a residue in the oral cavity and give rise to a transient malodor. Physiological halitosis is defined by the fact that the symptom, the malodor, disappears when oral hygiene is performed or with adequate hydration. And conversely, pathological halitosis is where the symptom is caused by pathology, and it is characterized by malodor that does not disappear with casual methods that would eliminate physiological halitosis. The vast majority of cases of genuine halitosis are attributable to intraoral phenomena, and this is known in the literature most frequently as oral malodor. The remaining percentage of cases that have a cause that is not within the oral cavity are known as extraoral halitosis. Intraoral halitosis, depending upon which studies you read, represents roughly 80 to 90% of genuine halitosis cases, and subsequently this has been the most widely researched area of this field. The pathogenesis here is related to production of volatile sulfur compounds by putrefaction of food debris, salivary proteins, and desquamated epithelial cells by mostly anaerobic bacterial species. The main loci of this microbial activity is the posterior dorsal tongue, and also within gingival crevices and periodontal pockets. Saliva itself can also act as a reservoir of hallucinogenic bacterial activity. The many other causes of oral malodor are usually fairly obvious, for example oral sepsis, and these should be easily detectable by general practitioners. Many medications cause dry mouth or xerostomia, and this can secondarily lead to an increase in oral bacterial activity, exacerbating or even creating the halitosis symptom. It is also known that intraoral prostheses can harbour populations of bacteria if they're not regularly cleaned. Extraoral causes of halitosis are in the minority, but perhaps more frequent than many clinicians would think. It has been a less well-researched topic compared to oral malodor, and there are many different conditions that can cause this symptom. The specialities involved would depend upon the exact cause. For example, ENT, gastroenterology, respiratory medicine, endocrinology. Extraoral halitosis can be further subdivided according to the location of the pathosis. So we could talk about upper respiratory tract causes and lower respiratory tract causes. And this is a short list of example diagnoses that could possibly be responsible for the halitosis symptom. However, it appears that the majority of cases of extraoral halitosis are caused by blood-borne halitosis, which we'll discuss in a moment. Gastrointestinal causes of halitosis is a controversial subject. Conditions such as Helicobacter pylori infection and gastroesophageal reflux disease have been investigated as potential causes. But it does appear to be a myth that GI conditions are a common cause for halitosis. In fact, some experts believe that it accounts for less than 1% of cases. So, what do we mean when we say blood-borne halitosis? Well, this is referring to chemicals present in the bloodstream, which are then transferred in gas exchange in the lungs, and are then perceptible on the expired breath. There are several reasons why these chemicals would find their way into the circulation. It is known that certain foodstuffs, such as garlic or onion, can lead to a transient presence of odor metabolites in the blood, and similarly with certain medications which can be metabolized to volatiles, which are then expressible in a similar manner. This is an infrequent side effect with most drugs. Systemic diseases involving insufficiency of major organ systems, such as pancreatic, renal or hepatic, organs which very simply would normally function to filter the blood, removing toxins. If they are not operating above a certain efficiency, then this may be manifest as the presence of volatiles in the bloodstream. A handful of metabolic disorders are also involved in causing blood-borne halitosis. The mechanism of blood-borne halitosis is as follows. As you all know, the body requires oxygen, so blood is pumped from the heart to the lungs to collect this, and at the same time it is depositing waste carbon dioxide, and this is known as gas exchange. Blood-borne halitosis happens when odorant chemicals are also exchanged, and they can become vaporized and held in suspension of the exhaled breath. There's not much evidence for this statement, but it does seem logical that gas exchange is the most efficient pathway for blood-borne volatiles to be excreted from the body. However, the blood circulates everywhere, and consequently the volatiles may be expressed via different excretory pathways. Although the term blood-borne halitosis implies that only the breath is affected, it's more accurate to take into account these other excretory pathways. And so wherever there are volatiles present in the blood, there may be other malodor symptoms, such as body odor. We can come up with some theoretical criteria for any particular chemical to be able to cause this symptom. Firstly, the volatiles must be able to enter the circulation. The usual route is via absorption from the GI tract into the portal system and onto the liver and its hepatic enzymes. This is known as first-pass metabolism. So the volatiles must be able to survive this metabolism, and this can happen either because they are present in such quantities that they overload the enzymatic pathways, or because the pathways are compromised and a mismatch is created between the enzyme's efficiency and the workload that it is being subjected to. Perhaps most importantly, the volatiles must be stable in blood. Many compounds break down in blood, binding with transport proteins, for example. And usually blood-stable volatiles are neutral molecules. And a further requirement would be for the volatile to be capable of being transferred to the exhaled breath during gas exchange. As I mentioned before, there are only a handful of known metabolic conditions that are implicated in causing volatiles to be present in the blood. And of these, trimethylamine is the most widely researched. However, anecdotal evidence seems to suggest that the majority of patients with a suspected metabolic malodor condition actually test negatively for TMAU. And furthermore, the classical description of TMAU is of a fish odor symptom, but the mainstay complaint among this patient group is of a feculent odor. Around 70% of patients will complain of this, compared to only about 30% complaining of a fish odor. If we were to consider a group of patients with a suspected blood-borne malodor condition, we could make 4 divisions. The first group of patients we could call classical TMAU. They test positively for TMAU, and their odor symptom is the classical fish odor. Unfortunately, these patients have a variable response to the protocol, with some achieving better symptomatic relief than others. The second group I would describe as atypical TMAU. Although these patients are testing positively, they do not complain of the classically described fish odor. Instead, most often they are affected by a feculent odor. Again, there seems to be a variable response to the protocol. The third group we could call TMAU, but not TMAU. Although these patients have similar symptoms to the above groups, they are repeatedly testing negatively. And this is somewhat of a diagnostic dead end, given the lack of research on the other conditions. Dimethylglycinuria is a even rarer condition with similar symptoms. Patients in this group often adopt a protocol similar to the TMAU protocol, and strangely this seems to help some, which is hard to explain. The remaining patients would comprise unknown conditions, testing negatively for TMAU and unresponsive to that protocol. If we just quickly jump through another few definitions, so I've been using this term volatile. You may have also heard the term volatile organic compound. So this is referring to a carbon-containing compound with a high tendency to vaporize. And there are several definitions in use. Perhaps the most widely used one is a compound fulfilling the above attributes with a boiling point below 250 degrees Celsius. It's worth noting that some odorants do not contain carbon, for example ammonia or hydrogen sulfide, and are therefore not, strictly speaking, volatile organic compounds. A volatile sulfur compound is, as the name suggests, a sulfur-containing compound which has a high tendency to vaporize. If a volatile sulfur compound contains carbon, it can be termed organosulfur. But again, some volatile sulfur compounds do not contain carbon, for example hydrogen sulfide. And the definition of an odorant is quite simply a chemical that is able to activate the olfactory system. Odorants have three common characteristics. Firstly, they must be volatile to enable them to be transported in the air to the parts of the nasal cavity where the olfactory receptor cells are housed. They must be either water or lipid soluble. And lastly, they must be present in great enough concentration to trigger the olfactory chemoreceptor cells to send a signal along the olfactory nerve. And each odorant volatile has its own unique threshold level above which it is perceptible by humans. If we just look at the molecule dimethyl sulfide in a little more detail. So it's made up of a sulfur atom covalently bonded to two methyl groups. Dimethyl sulfide is a neutral molecule, making it stable in blood. So it is able to exist in the bloodstream without being broken down, and it's also able to be transferred to the exhaled breath during gas exchange. And this is in contrast to other volatile sulfur compounds such as methyl mercaptan and hydrogen sulfide, both of which are unstable in blood and are therefore not implicated in bloodborne halitosis. As we mentioned before, it appears that dimethyl sulfide is the most common volatile implicated in bloodborne halitosis. The in vitro character of dimethyl sulfide is varyingly described as unpleasantly sweet, cabbage-like, or similar to wild radish. It is known that it becomes increasingly unpleasant to judges as the concentration is increased. Dimethyl sulfide is a known irritant, and the various threshold values are listed here. And essentially, the point to take from these values is that dimethyl sulfide is perceptibly unpleasant at exceptionally low values. There are several possible routes of dimethyl sulfide production in mammals. Usually it is broken down from dietary precursors in plants. Methionine can be transaminated to produce dimethyl sulfide, and successive methylations of hydrogen sulfide and methyl mercaptan will also produce dimethyl sulfide. Dimethyl sulfoxide can be reduced to produce dimethyl sulfide. We'll now briefly discuss the behavior of dimethyl sulfide in physiological systems. So, some bacterial species known to synthesize dimethyl sulfide have been demonstrated in the human GI tract. Indeed, dimethyl sulfide is a component of the characteristic odour of feces inflatus, along with hydrogen sulfide and methyl mercaptan. These sulfur compounds are likely the result of intestinal bacterial activity. However, it's been shown that of all the volatile sulfur compound gases produced in the colon, over 90% are reabsorbed by specialized lining tissue rather than being emitted. Hydrogen sulfide and methyl mercaptan are broken down in this manner, but it appears that dimethyl sulfide is not metabolized here and it passes much more easily into the bloodstream. Dimethyl sulfide appears to be a substrate of both cytochrome P450 and flavin-containing monooxygenase. It's subject to renal and pulmonary excretion. That is, it's secreted in the breath and the urine. And a study was also able to show that dimethyl sulfide was present in cow's milk after their diet had been loaded with either dimethyl sulfide or methionine. Following a recent review paper about physiological concentrations of sulfur compounds, much more is now known about the normal ranges of physiological dimethyl sulfide. What is clear is that these levels remain very low in the absence of any specific pathology. At this point we can introduce a new term, Dimethylsulfidemia, which could be defined as pathologically raised levels of dimethyl sulfide in the blood. We could also talk about pathologically raised levels of dimethyl sulfide in the urine, and this could be termed dimethylsulfiduria. There are several known causes of dimethylsulfidemia, and therefore again it is a symptom, or rather a sign, as opposed to a diagnosis. And it's helpful if we consider the differential diagnosis of dimethylsulfidemia in these same categories as we used earlier, as for blood-borne halitosis. So, starting with metabolic, hypermethylanemia is a rare condition. It's got several known causes, and some are quite serious syndromes presenting at birth. Methionine is a essential sulfur-containing amino acid. It's normally metabolized in a pathway resulting in cysteine, and it's quite a complicated pathway. There are several different enzymes involved, and there are many intermediary stages. At this point we need to recall that dimethyl sulfide is a transamination product of methionine. Consequently, if there's insufficiency of the methionine pathway, then the alternative transamination pathway is used. And so, where there is hypermethylanemia, we will usually see an accompanying dimethylsulfidemia. One of the most recently discovered causes of hypermethylanemia is isolated persistent hypermethylanemia, and this has been attributed to deficiency of the enzymes MAT1 and 3. These two enzymes normally catalyze an important stage in the methionine pathway, converting methionine to an intermediary compound. And this cause of hypermethylanemia is of interest because, as the name suggests, it's a isolated feature. There's no other symptoms. The other known metabolic condition that causes dimethylsulfidemia is in the process of being described, and we are waiting for this research group to further define the exact nature of this metabolic condition. This same paper found that dimethyl sulfide was the most common volatile involved in blood-borne halitosis, although this was based on a small sample. But if the figures are representative of the general population, then the prevalence of this new metabolic condition could be estimated at 0.25 to 1.25%. This could be contrasted with the heterozygote TMAU instance, which was lower. And heterozygote TMAU status does not necessarily imply symptomatic TMAU. Many of them will be carriers. If we move on to systemic conditions that can cause dimethylsulfidemia, then the only condition to meet this description is fetor hepaticus. This is the odour produced mostly on the breath of patients with liver failure. The usual pathogenesis here is portal hypertension, so blood is not taking the normal route from being absorbed from the GI tract through the liver. This fetor was traditionally attributed to ketones and ammonia. However, in the case of ammonia, it has been demonstrated that this is not the case because it is not liberated from solution at physiological pH. And rather than ketones being the main contributor to the odour, it's in fact dimethyl sulfide. Several papers have shown that breath dimethyl sulfide is markedly elevated in patients with fetal hepaticus, and in vitro experimentation with solutions of dimethyl sulfide has been reported to produce an odour very similar to that of fetal hepaticus. Interestingly, one paper reported elevated trimethylamine levels in the blood and urine of patients with fetal hepaticus. However, again, this was not thought to be the greatest contributor. Medical textbooks describe the odour of fetal hepaticus with quite varying terms, including musty, mousy, or even sweetly fecal. One description even likens it to the odour of dead mice. Since dimethyl sulfide is the greatest contributor to the odour of fetal hepaticus, it's logical to assume that the in vivo character of dimethyl sulfide is similar. In terms of foodstuffs that could potentially create dimethylsulfidemia, very little is known on that subject, but it does seem that in the absence of any metabolic condition, dimethyl sulfide would not be elevated through diet alone. Finally, if we move on to medications that can cause dimethylsulfidemia as a side effect, dimethyl sulfoxide can be used as a therapeutic agent in its own right, but most usually it's used as a pharmacore, so other drugs and functional groups get attached to the molecule. This is usually to enhance their penetration of skin, because dimethyl sulfoxide is a very good agent for topical administration. A known side effect of dimethyl sulfoxide is a garlic taste in the mouth, but the relevance to dimethyl sulfide is unknown. Dimethylsulfidemia results because dimethyl sulfoxide is metabolized to, amongst other things, dimethyl sulfide, and it's subject to renal and pulmonary excretion. And only 2 other medications are known to have dimethylsulfidemia as a side effect, although they are much less widely used than dimethyl sulfoxide. Dimethylsulfidemia and the previously mentioned new metabolic condition offer a number of new research opportunities in the field of bloodborne halitosis. The standard laboratory method for detection of volatile sulfur compounds is by gas chromatography. A portable machine has been developed called the oral chroma. This machine is regarded by some as the gold standard. It's particularly useful in differentiating between intraoral and extraoral halitosis. What is already known is that there is a close correlation between blood dimethyl sulfide and breath dimethyl sulfide. However, the exact level of blood dimethyl sulfide required to produce the halitosis symptom is unknown. And it would also be useful if dimethyl sulfide urinalysis could be demonstrated to be a valid screening tool for dimethylsulfidemia. And finally, I would like to again draw your attention to the fact that MEBO research is currently carrying out a volatile organic compound breath analysis research study. And specifically, we are most interested in recruiting participants with atypical TMAU symptoms and those who are not testing positively for TMAU.